Hello, everyone, and welcome back to the NWR Virtual Healthcare Day for November 2021. My name is Finn Blake, and up next this afternoon, we have Tissue Repair. Tissue Repair is a clinical stage biopharmaceutical company developing advanced wound healing products for chronic wounds and the aftercare of cosmetic procedures, with the potential for further development of related technologies. The company is in the final stages of completing its phase 2 program for its chronic wound product, TR987, and just yesterday announced the progress on its core objective of entering and conducting a phase 3 clinical trial for this product. This afternoon, I've got the co-founder and executive director, Tony Charara, joining me. And a little bit later on, he will be joined by Daryl Reid, the COO. If anyone in the audience would like to submit a question for the Q&A session at the end of the presentation, then please do type it in the Q&A panel using your screen at any time during the presentation. Without further ado, I will hand it straight over to Tony.
Oh, great, thanks very much. A little bit of background. I am a co-founder of Tissue Repair and got involved in the company in 2013. It actually at that point had done a phase 2A trial and got really promising data on chronic wounds and basically fell in love with the data set at that point. So as a bit of history, we did a restart off the back of that and basically set out to get a second read on clinical efficacy with 2 separate phase 2B trials that we recently completed in 2020. The company was actually going to get listed, or the technology was going to get listed on NASDAQ, had about a 200 mil valuation back in 2007. We've essentially taken it a step further by getting a second clinical read on efficacy. And I'll talk to that in a minute. I think what excites us about the technology is its platform. So I'll talk to it in a little bit of a minute. But it's essentially a yeast. We take out all the proteins and the lipids. We place that cell inside an open wound, and as a result of that, the body perceives it as an infection, and it kickstarts the whole cascade of wound healing process. Importantly, it's a mechanism of action that's applicable to any drug, applicable to any animal or any human, and can be used for a wide range of wounds. I think the real power of the company is its data. So we've been in the clinic in about 200 patients. We've actually finished the clinical trial work on our phase 2 trial. And we showed about a 20% difference in incidence of complete closure in that trial. No one's got a drug label in chronic wounds for about 25 years. And no one's actually shown that level of efficacy at a phase 3 or at a drug product. So the fact that we got that in a sort of a double-blind randomized clinical trial provides really strong foundations going into phase 3. The other thing that really excites us is the fact that we've just got this consistent measure of efficacy. So if you look at the phase 1, phase 2A, or the phase 2B trials, at each point you're getting a similar signal of efficacy. So it's really great data foundations going into phase 3. I think a lot of people, as I said, there hasn't been a drug approved in 25 years in this indication. Most companies go down a medical device path. So if you actually achieve that label, many people consider it a massive prize. And we're not at phase 1, we're not at phase 2A. We've essentially finished our phase 2 clinical work, and we're preparing for phase 3 entry. So we've recently done our IPO, which we closed. Nothing's changed since the prospectus that we've lodged, albeit the announcement that we made about progress with respect to manufacturing. But we're planning on entering into, we've got enough capital to essentially get approval to go into phase 3 and conduct a phase 3 trial. So although our main prize is this drug label, that's where we feel the most value is going to get created. We've got this near-term secondary strategy of commercializing a cosmetic product. And we did a phase 2B trial in an aesthetic indication and basically showed close to double the skin quality effect. And that was statistically significant in a double-blind placebo-controlled trial. We don't need to go down a drug path for that. We can launch that product straight away. and our path is to essentially start a real-world clinical trial in the first half of 2022 and go straight into a commercialization of that product thereafter. So I think I've talked a little bit about this, but the benefits, I think, are it not only accelerates healing, but it affects closure in wounds that are stalled. And the reason why it does that is, you know, when you introduce this inflammatory agent, the body essentially goes back to baseline in wound healing. There's essentially a Trojan horse or an infection, and that starts the whole cascade of wound healing from baseline. And that's why we're seeing the closure rates, and that's shown up in the evidence. So we showed about a 20% difference in complete closure with a p-value of 0.1. That's at 67 patients. In a phase 2B trial with that number of patients, that's a real strong signal. So, you know, in phase 3, we're essentially powering that study up to about 300 or 350 patients. And all we're seeking to do is replicate that signal. We're not seeking to accelerate it or improve the efficacy signal. We really just want to run the trial that we ran in phase 2 and change, without changing too much around that protocol design, with a greater cohort of patients to drive that p-value down to 0.05. And all our analysis, our statistical analysis suggests that that'll occur at around 300 patients. I'll get Daryl, Dr. Daryl Reed, who's our COO, to quickly talk you through the mechanism of action here.
Thanks, Tony. Look, at a cellular level in wound healing, it's a really complex process. There's a lot of different things going on with different cell types, growth factors, cytokines. But where we come in is right at the start of that. So we impact macrophages. We know that they're the key cells that make up our first line of defense. They also stimulate other immune cells, but they themselves need to be activated. And the way that happens is through receptors that are located on their cell wall. So over time, macrophages have adapted to respond and recognize to fungal pathogens and also bacterial pathogens. And that's why fungi or yeast can stimulate macrophages. We know it's a specific polymer in that fungal cell wall. It's called a beta-glucan, and that's what stimulates the macrophages. So in the same way that a lock and a key fit together, that beta-glucan on the yeast fits into receptors on the macrophage surface. So our development product is TR987. It's a non-infective beta-glucan molecule, which, as Tony said, it's had all the lipids and proteins stripped out of it, and it's just the skeleton remaining. That beta-glucan is what we've called glucoprime. That's our active ingredient, and that's the platform technology that underpins what we're about to embark on. So studies have shown that TR987 is really able to behave just like a fungal pathogen. Given that the last drug approved for chronic wounds was Regranax back in 1997, we're really confident there's an opportunity for a new entrant like glucoprime that is supported by robust clinical evidence. And the fact that some of the evidence which has been published on that mode of action that I just walked you through actually used glucoprime provides us with a high level of credibility. There's a number of key points of difference between the current used therapies and what we offer through glucoprime. But a couple of key call-outs are that most current therapies are derived from placental tissue. We know that's expensive. It's often challenged by inherent product variability. Glucoprime, in contrast, is far cheaper to produce and can be reliably purified. We've got a really strong safety profile, having been tested on more than 200 patients with no significant adverse events. But for me, the really key thing is when you think about chronic wounds, often requiring complicated grafts, need to be applied by a clinician. What we're offering here is a product that has the advantage of being able to be applied topically directly onto the wound without any concerns about infection. It can be stored at room temperature, and when you wrap all that up, what it means is that people who, for example, might be living in an aged care facility can be treated in that facility. They don't need to be making daily or weekly trips into the hospital. This slide here gives you an idea of the potential applications of glucoprime. Tony alluded to this at the start, but if we look at the first column, glucoprime's our API. Underneath that, you've got the TR987 gel. That's the product we're taking into chronic wounds phase 3 clinical trials. Beside that, you've got burns and surgical and other complex wounds. Both of those represent potential future markets. Other dressings and matrices are things that are currently used, and there's no reason why glucoprime can't be used with those products. TR Pro Plus is exactly the same product as what TR987 is, but that's the one that's geared to our aesthetic product. And that's the one we're preparing for commercial launch next year.
So I'm probably just going to skip a few of these slides here. We'll make this pack available after, but there's a couple of key points that I just wanted to make sure that they resonate with me in terms of this. I'm escrowed for such a long period of time. I'm a big believer in the company. And just to give you a sense of how I sort of look at value, but that chronic wound label is our main driver of shareholder value. A lot of people consider that a massive prize if you achieve it, even if you're at phase 2 or just starting a phase 3 in an indication with this much TAM. The market values for these companies are huge. While we're doing that, we're trying to basically, really as a hedge, commercialize that aesthetic product as a secondary focus. I think there's a bunch of announcements that, or milestones, if you like, that are going to come out over the next sort of medium term. But we're aiming, we've got a program of work to basically get phase 3 approval that is progressing over calendar 2022. We're hoping to enter phase 3 trials towards the end of calendar 2022. And while we're doing that, there's a real world commercialization of a real world clinical trial of the gel, which we'll hopefully can announce the feedback on in the first half of 2022 as well. We've already done our market research report. We're getting really great feedback as per that announcement this morning or yesterday around the physician's attitude to the product. And I think the key thing, one of the key dependencies in getting into phase 3 is manufacturing. So the fact that we've manufactured 2 pilot batches of API since we've done the pre-IPO round and the IPO, and that material is consistent with the reference material, the material used in the phase 2 trial. That's a big milestone for us, because it basically means that one of the big risks of getting into phase 3, look at that risk curve, has been significantly reduced, because a lot of our phase 3 dossier is around CMC. The actual protocol is the most innocuous part, because we're just going with a mandated FDA endpoint. I think this is the real power of the company. It's its data. So not a lot of companies have double-blind, placebo-controlled data signaling efficacy. If you look at the device space, or drug quality data, and this is really what got me involved in the company 7 years ago. And I didn't have the benefit of the phase 2B trial. I had the benefit of the phase 2A data. And the fact that you got a second read on that, so you've got essentially 2 double-blind human trials close to 10 years apart, over 10 years apart, which evidence efficacy, is a real strong foundation going into phase 3. So you can sort of see that middle group is a 20% difference in incidence of complete closure. Most clinicians would say a 10% difference is clinically meaningful. That intention to treat group is based on the gold standard product in market at the moment. That's essentially reimbursed at about $1,000 per application. So you can sort of see we're comparable, if not superior to that product. But the real power of TR is its data. So is its cost. So because our starting material is yeast, it costs us about $3 to essentially manufacture a 15 ml tube. By contrast, Regranax is reimbursed at maybe $1,200 for a 15 ml tube. So that cost, combined with this measure of efficacy, combined with the fact that it's a gel, because that data there on the right is essentially a device, it's a patch. That matrix of benefits, we feel really provides a compelling product that's that significantly has a significant competitive advantage compared to what's available in the market. This is a wound we treated in phase 2B. This was out of the repatriation hospital in Melbourne. That wound there was present for close to 4 years, and we managed to close that in 10 weeks. And the physicians were actually, unblinded by this. They could actually tell which was active and which was placebo, because this was a patient known to them. And that's an atypical closure outcome that we achieved in the trial. This just compares the signal of efficacy across phase 2A and phase 2B. And this is on an endpoint called percent wound area reduction. And the only point here is the consistency of the signal across 2 separate trials. So that's what we're excited about going into phase 3, because we sort of feel we've got good foundations to be able to replicate the signal that we had in phase 2B in phase 3. Again, typical example of wounds. These are Australian patients in the 2A trial, and the closure or healing that we evidenced at week 4 and week 12. And these 3 things are really what we need to get phase 3 approval. Like, once we get approval, that's a massive milestone for the company and another catalyst we feel for value. And A is the big one: analytical development, product manufacture. So the fact that we've manufactured fresh pilot batches, it's consistent with the API we've used, gives us a lot of confidence that we get that we've de-risked that work stream. The phase 3 trial, or the protocol, is really uncontroversial. We're not pushing the limit on that. The FDA have given us guidance around what they'll accept. It's complete closure. That's the core gold standard endpoint, and we're not trying to change that attitude. We're just adopting it. So we sort of feel A is really our key risk area in terms of getting into phase 3. I'll get Daryl to quickly go through the cosmetic product. Again, it's a secondary focus, but again, the roots of the company are data, and here we've got some really compelling data that Daryl can talk you through.
Look, I'll keep it pretty quick, but basically what this trial was looking at was skin quality, and it was in patients who had undergone a laser ablation, which essentially heats up and destroys a layer of skin. So it's almost like a burn. We applied the product for 5 days, and then at 28 days, we assessed the skin quality, looked at 2 measures, elastosis and wrinkling. On the left, you can see the difference in elastosis in the TR987 patients versus the placebo. That was a 40% absolute difference at a p-value of 0.011. Wrinkling on the right-hand side, a similar improvement, 35% at p equals 0.04. So both measures showed a statistically significant improvement in skin quality following that laser ablation.
Yeah, and just to add, you benchmark every other post-procedure topical, and no one's shown this kind of evidence. We've done a lot of that work. So again, we could go into phase 3 with this. We could get a drug label around the aesthetic product, but we've chosen to launch it as a cosmetic because the patients pay. It's a bit like Botox. So to give you an example, someone might spend $2 to $5 grand in the US on a laser procedure. It's about 3 months to get the full benefit of the skin quality. The proposition to the patient is to essentially, for an extra couple of $100, you achieve the full benefit or accelerated healing as measured by skin quality at day 28. You're basically doubling the skin quality benefit you see. So we feel that's a pretty compelling proposition. So again, though, this is a secondary focus for us. We're not really taking our eyes off that main prize, and all the resources of the company, or the majority of them, are geared towards phase 3 approval and conducting the phase 3 trial. I think there's only a couple of other sites. I've talked about CMC and IP. So I've talked about CMC. IP, 2 core areas here. We are considered a new chemical entity by the FDA. No one's got approval with a product based on our molecule, which is in the beta-glucan family. So as a result of that, we get 5 years of exclusivity on marketing approval, and we get 10 years in Europe. We've also lodged a fresh patent in June. That's powered by all the clinical data you've seen here, which has never been published up to that point, and all the characterization work. And we feel we've got good prospects, and that patents the molecule itself. It's not a process patent. So we feel we've got good prospects of a further 21 years of protection around that. Really quickly on the board of management, look, this is a great board. Like, you've got Max Johnston, who was the MD of Johnson & Johnson for many years. He was also on the board of PolyNovo. He actually left the PolyNovo board and joined ours, and recently he's now the acting CEO of PolyNovo. Professor Craig Stamp was at Allergan, launched Botox in the mid-2000s, and also was the managing director of Bausch & Lomb, and he was also the general manager of Device Technologies, which sold a huge amount of product in that aesthetic space. And then you got Jack and Brian on governance, and so Jack's out of Hunter Hall, and Brian Gray was a managing director at JP Morgan, so they're on finance and governance. Again, I think we've got, I'm pretty proud of the team we've got here as well. Like, Bill Boost actually manufactured Regranax. So you've got someone on CMC that's manufactured a wound product, let alone a topical. He also manufactured Santinol, which is the other largest topical product that's sold. It's a debriding agent. Mark Waring on clinical affairs and reg, and he was actually at the University of Sydney and did his honours work around beta-glucan, which is where the original research started. And you've got Pramod Nador on analytical chemistry and controls, and he actually manufactured our phase 2B clinical supplies and was instrumental in getting approval around CMC with the FDA. So, and Daryl, he's given an overview. And then likewise on the advisory panel, Professor Vaclav there at the end is considered a global expert in beta-glucans. He's written the most peer-reviewed research around beta-glucans. Professor Woodward is a big believer in the technology. He was the PI on the 2A trial, and 13 years later was a PI on the Australian sites in the 2B trial, and a big believer in the evidence that he's seen in the use of the product. And Dr. Thomas Serena is considered a global expert in chronic wounds and running clinical trials in chronic wounds. So that's the team. The only other thing I would want to say is around Escrow, and just wanted to reconfirm, all the seed shareholders and the founding team, which is about 55% of the register, are escrowed. And they're escrowed. I'm personally escrowed for 30 months. Everyone's escrowed day one, and 80% of their holding is escrowed for 24 months. So that's a really strong escrow period. And a lot of that is around our belief that we've got a shot and aligning our interests with the shareholders in terms of the real value here is around that program of work to get a phase 3 label. So I just wanted to reinforce that. I'm seeing a lot of questions around share price movements. I have, I'm unaware as to why the share price post IPO has moved like that. There's been, the prospectus is valid. We placed an announcement out to the market yesterday, which basically gave updates around where we're going. And as far as the company and the board is concerned, the prospects of the company, are as detailed in the prospectus, and the opportunity is as valid and hasn't changed as when we ran the IPO process. So I guess with that, I'm happy to take questions.
Unfortunately, we've kind of run out of time a little bit, Tony and Daryl, but I'd love to just ask one to just wrap up very quickly. What's the main reason that both of you are very confident in tissue repair as we approach the new year? I know that you mentioned a lot of unique angles that is unique to tissue repair, but what's the one that you're most excited about?
Well, I can start. So for me, it's data. Like, you got a really strong signal at phase 2B on a 20% difference in complete wound healing. If you achieve that in phase 3, that's exceptionally clinically meaningful, we believe. And no one's achieved a drug approval in 25 years, let alone achieved that kind of difference. So that data and the consistency of the signal, right from when I first got involved to the data that we achieve now, like, it's that profile is what, giving me a lot of confidence around our ability to get a phase 3 outcome.
For me, just to build on Tony's, he's exactly right. But, we're not looking to change anything in phase 3. We're simply replicating what we got in phase 2 with a bigger group of patients. So all that risk of change is moving from 2 to 3 is non-existent. And the final thing is just our secondary stream in the aesthetic. That's something you don't often see in a biotech, so that excites us as well.
Beautiful. Tony and Daryl, thank you so much for your time today.
Good one. Thank you.